Thank you, Rinku, for your kind introduction. Greetings of the day to all my teachers and anites. I deem it an honor to be a part of this alumni lecture series. Being a teacher by profession, I am really fortunate to deliver a talk on the eve of the upcoming Teachers' Day. Teachers' Day is a very memorable day when all of us remember all our teachers who taught us, who trained us, and gave us a direction to carve out our future career paths. I wholeheartedly acknowledge the contribution of all my teachers with specific reference to Sister Preside, who trained us to be systematic, precise, focused, and analytical. Above all, she has kept all of us in her daily prayers. I also wish to acknowledge my PhD guide, Professor P.V. Subbarao at IISc, who gave me the freedom to think and set an impetus to my research career. It's my tenure at IISc that really gave me the confidence to pick up research and teaching as my career. I'm passionate about biology, more so about immunology. And when Vasavi approached me to deliver a technical talk, the most appropriate topic post-COVID was to highlight the role of our immune system. COVID in one way stressed upon hygiene, precautionary measures, and the importance of vaccination to combat diseases or infections. So today's topic would be self-defense, the power of our immune system. I'll try not to be too technical, but please feel free to stop for any clarifications. So I'll just share my screen and then we can go ahead. So to begin with, let's define immunology. Immunology is the study of the immune system, the defense mechanism of our body. It is the study of the immune cells and organs which help us to protect against infection by a variety of organisms. It could be a virus, bacteria, a fungal organism, a parasite. It could be a toxin or a carcinogen. Now, the word immunity is derived from the Latin word immunis. Immunis means exempt. In other words, immunis offers a state of protection from infectious diseases. If you see this slide, you will see that there are a variety of pathogens which can infect us. So it could be toxins, it could be fungal organisms, it could be bacteria, it could be parasitic organisms, it could be pollutants, which are toxins, it could be carcinogens, which can cause cancer, and it could be viral particles. All these are non-self, that is, not part of our self. And our immunity will resist infections caused by these factors. Now let's go a little into the historical development of immunology. Edward Jenner was known as father of immunology. It was his keen power of observation that actually gave rise to the concept of vaccination. It was a time when smallpox was in vogue, it was a contagious disease, and it was even fatal. So it was his keen observation power that helped eradicate smallpox across the globe. He noticed that milkmaids who were working with cows contracted the mild form of cowpox. All those who got the mild cowpox did not get smallpox. So this led to the process of vaccination. Edward Jenner took the fluid from the cowpox pustules and infected healthy individuals. These healthy individuals, they contracted the mild cowpox but never got smallpox. And this is how the process began. Later, it was the work of Louis Pasteur who initiated the process of vaccination. Louis Pasteur was working on cholera, and the causative organism is Vibrio cholerae. So he isolated Vibrio cholerae, and then he had to go on a vacation. After a month, he came back and he took the sample from the refrigerator and tried to do his experiments on experimental animals. To his surprise, he found that all these animals had mild symptoms of cholera, but they survived. So then he observed that if you keep the pathogen in unfavorable conditions, then they lose their power to infect. So he then came up with an idea that you grow the bacteria in unfavorable conditions, or you kill them by heating them, or by treating them with chemicals. So these are incapable of causing infection, but when you inject them into healthy individuals, they are immune to these particular infections. So in honor of Edward Jenner, who worked on cowpox, in Latin, cow means vacca, and therefore he called this vaccine and the process vaccination. He was successful in developing vaccines against anthrax, rabies, and so on. So thus all branches begin with a concept and then end up in applications. Immunology is the only branch of immunology, of biology, where the application was identified first, and then immunologists start looking at the concept behind the immune response. So the concept of immunology revolves around 3 R's: recognition, response, and remembrance. So what do I mean by recognition? The immune system recognizes pathogens. So they should be able to differentiate self from non-self. What do I mean self? Self means anything that is part of your body. Non-self is anything out of your body. It is foreign to the body, and therefore it is called non-self. Once the immune system recognizes it as non-self, or a pathogen, or an enemy, then it responds. So the second R is response. So it responds by killing the pathogens. The beauty of the immune system is it remembers. So when the immune system sees a pathogen, it recognizes it, it responds, kills the pathogen, and has immunological memory. That is the 3 R's which constitute the concept of immunology. Now let's go on to the types of immunity. Now, immunity is of 2 types: innate immunity and acquired immunity. Innate immunity is natural immunity. It is the first line of defense, whereas acquired immunity is the second line of defense. It is also called adaptive immunity. Adaptive immunity gets activated only when it encounters the pathogen, but innate immunity is always present. A simple analogy. Now, innate immunity is like the border security force. The border security force role is to protect the borders from the entry of enemies. And that is what the innate immunity does. It prevents the pathogens from entering the body. And it is always equipped. By chance the pathogen enters, then the components of the innate immunity kill them. Meanwhile, innate immunity sends a signal to the specialized armed forces, that is the adaptive immunity. So they tell them there is a pathogen here, you better gear up and then try to eliminate the pathogens. So adaptive immunity takes some time because it wants to understand the pathogen. It analyzes it. It designs a strategy how to kill these pathogens. And therefore, innate immunity is always there. Its main role is to prevent entry, whereas adaptive immunity, it recognizes it, devises a strategy to kill the pathogen. Most important, it remembers. It has immunological memory. So when it sees the pathogen for the second time, it knows, yes, I have seen you before. I know you. I know how to tackle you. And that's how, because of immunological memory, the second response is going to be much quicker, much more stronger, and much more effective. On the other hand, innate immunity does not remember. So every every time it sees the enemy, it only recognizes that non-self response. So the response for the first and the second time always remains the same. So if you look at this graph, you will see the difference between innate and adaptive immunity. Now, as you can see here, innate immunity begins within hours after the exposure to the microbe. Then it sends a signal to adaptive immunity, and the adaptive immunity now begins several days after the innate immunity, and then it remembers. Now, look at the other part of the graph where the uh, individual is exposed to the same microbe for the second time. Innate immunity again starts acting within hours, and the response is more or less the same. Whereas adaptive immunity, on the other hand, since it already knows the pathogen, it the response is quicker, it is much more stronger and much more effective because it has immunological memory. Whereas innate immunity response remains the same whether it meets the first time, the second time, or the third time. Now we'll slowly move on to the components of the immune system. Now, as I told you, innate immunity prevents entry. Now, the skin does not allow any pathogen to enter. So it is a physical barrier. However, we have three major systems: the respiratory system, the gastrointestinal system, and the urogenital system, which is exposed. So if all these three systems are lined by a mucosal lining, now let us consider the respiratory system. In the respiratory system, say the germs enter through the nose. We have nasal hair and we have mucus to trap the pathogens. Say the pathogen enters into the airways, which lead to the lungs. You have hairy like structures called cilia, which move upwards and pushes the mucus entrapped pathogens via cough reflex. That's how we are able to throw mucus along with the pathogen. Say we ingest food which has pathogens with it, the first step would be to meet the highly acidic gastric juice. Many pathogens cannot tolerate the acidic pH of the stomach and they do not survive. But certain acid resistant bacteria move down into the intestine. There, as I said, the intestine is lined by the mucosal lining. It does not allow the pathogens to stick to it. Then there is competition from the good bacteria, that is our gut bacteria. So there is a tough competition between the good bacteria and the bad bacteria. The good bacteria does not allow the bad bacteria to attach. And second, the bad bacteria doesn't get enough food, and therefore it doesn't stick and penetrate to cause infection. And we have other mechanisms like in the eye, you have tears which wash away the pathogens. It also has some enzymes which can degrade bacteria. Then we have urine which washes away the pathogens in the urogenital tract. So we have chemical barriers like the acid pH, high temperature, and enzymes. And we have physical barriers like the skin and the mucosal lining, which prevents entry of the pathogen. However, if the pathogen is successful in entering the body, then we have cells which can fight against these pathogens and kill them. The most important ones are the neutrophils and the macrophages and the natural killer cells. On the other hand, we have adaptive immunity, where there are two types: the humoral response mediated by B cells, which will produce antibodies. I think most of you would have heard antibodies. They are the magic bullets that are produced by the B cells. Then we have the cell mediated response mediated by the T cells, where you have two types: the helper cells and the cytotoxic cells. I'll come to that in detail a little while later. Now, what connects the innate immunity to adaptive immunity? Remember, I told you that innate immunity is the first to attack, but it sends information to the adaptive immunity to gear up to fight against pathogens. So the cells which mediate between the innate immunity and adaptive immunity are called dendritic cells. Macrophages also do this role of linking the innate immunity and the adaptive immunity. Now, if the pathogen enters, what does the innate immunity cells do? I'll come to that in detail a little while later. As I told you, macrophages play a very, very important role. They are the scavengers. They engulf the pathogen. As you can see, they have pseudopodia-like projections. They recognize the pathogen, they engulf it, and within themselves, they digest it and kill the pathogen. This process is known as phagocytosis. As you can see in the right, you can see the macrophage with its pseudopodia engulfing and trapping the pathogens. Now, there is another mechanism of innate immunity, that is inflammation. Say there is a cut on your skin. What happens? Pathogens start entering inside. Now, how does the immune system know where the infection is and that the pathogen has entered? Now, cells talk to each other. As I told you, you have neutrophils moving across in the blood vessels, you have monocytes, and you know that blood is continuously flowing. But as I told you, cells talk to each other, they have some communication, and because of some chemotactic factors, they identify the point of infection. They squeeze in through the blood vessels, and then they reach the site. And as I told you, the earlier mechanism, they engulf it and they kill the pathogens. However, how do we know that there is an infection? Wherever there is a cut, there is swelling, there is redness, it is warm to touch, it is painful. And therefore, these are the 4 cardinal signs of inflammation. Once the infection is subsided, then blood supply is restored there to that area and the scar tissue is formed. Thus, innate immunity has physical barriers to prevent entry, chemical barriers like the gastric juice in the stomach, then enzymes and the good bacteria, which can prevent the entry. And of course, we have phagocytosis and inflammation, which can take care of pathogens. Now we slowly move on to adaptive immunity. Adaptive immunity is more sophisticated because it has cells and organs. Now, the major organs are the bone marrow and the thymus. B cells originate from the bone marrow, and that is why they are called B. And T cells originate from the thymus, and they are called T cells. These 2 organs are responsible for the B and the T cell maturation. Of course, we have other organs like the spleen, the lymph nodes. You can see the lymph nodes like tonsils in the throat. Then you have lymph nodes in your armpits, in your loins, in your intestine. What do these do? The spleen and the lymph nodes, the tonsils, Peyer's patches, they form the meeting place. Now, say the B cell is in one place, the T cell is in the other place, the antigen is in one place, macrophages are in another place, then you cannot elicit an immune response. So all of them meet at organs like the spleen and the lymph nodes. In this meeting place, the B cells, the T cells, the antigen presenting cells, all of them ensure that they elicit an immune response and kill the pathogen. Now, coming to the cells, I think most of you at one point or the other would have given your blood sample for a complete blood count. So we have RBCs, that is a red blood cells, you have platelets, and the most important cells which participate in immune response are the WBCs or the white blood cells. So you have neutrophils, then you have monocytes, which are part of your innate immunity. Then we have the T cells, then you have the B cells, which have antibodies as its receptors. They are like antennae, which recognize the antigen, you have natural killer cells, and you have macrophages. All these are the white blood cells which play a major role in eliciting an immune response. Now, these are the cells. As I already told you, the B cells have the Y-shaped molecules or antibodies as antennae on the surface. Then you have 2 types of T cells. The helper cell, as the name suggests, it helps the B cell to secrete antibodies, and it also helps the cytotoxic C cell to kill viral infected cells. And of course, you have natural killer cells, and you already know the role of dendritic cells and macrophages in phagocytosis and connecting the innate immunity to adaptive immunity. adaptive immunity. So what is the difference between a B cell and the T cell? Now the B cell looks at pathogens which are outside the cells. They are moving all around the body through blood circulation. So B cells attacks only those pathogens or organisms which are outside the cells. Now on the other hand, the T cells attacks the pathogens which are inside the cells. How do you know how a virus attacks? It enters the cell, it starts multiplying, kills that cell, bursts that cell, comes out, infects another cell, there it multiplies, and the process continues. Now, how does the immune system know that the virus is inside the cell? So the T cell's job is to recognize such viral infected cells and then kill them. The T cell also recognizes cancer cells. Now, what are cancer cells? They are normal cells which change into cancer cells. So the cancer cell also has to tell the immune system, I was a normal cell, now I have become a cancer cell, better identify me and kill me. Say you have an organ transplantation. So any organ which is not part of you but comes from somebody else is again non-self, provided there is a perfect match. If it is no perfect match, again it tries to recognize the foreign cells as non-self and it can kill. Say for example, your blood group A, somebody gives you blood group B, which is not your blood group, and so the immune system tries to kill all those blood group B cells. So that is the job of T cells. B cells kill pathogens which are outside the cells, happily moving around all over the body. So again, as I told you, the T cells are of two types: the helper cell, which helps B cells. It also helps the cytotoxic C cell or the killer T cell to kill viral infected cells. Now let's look at the role of B cells. So I told you the B cell has antibodies as its receptors. These recognize specific regions on the pathogens. It is like the lock and the key mechanism. So let us take this example where a pathogen has three different types of projections. So there is one antibody which recognizes this, there is another antibody which recognizes this, and the third antibody recognizes another projection. All these are perfect fits. So when this antigen or pathogen enters, three different types of antibodies can pounce on it, bind it, and then it is engulfed by the macrophage. So each B cell produces only one type of antibody. Now, a little more in detail. So if you see there are two B cells, a pathogen which is light blue enters. Now this B cell recognizes it, whereas the other B cell cannot recognize it. I told you it is like the lock and key. They're extremely specific about the pathogen to which they bind. Once they bind, then they get help from the helper T cell, which gives a signal. And once the B cell gets the signal from the helper T cell, it becomes huge. It starts producing millions and millions of antibodies. Remember, a bacteria does not walk into the body alone. There are millions of bacteria which enter inside, and therefore the immune system requires millions of antibodies which recognizes that particular pathogen to neutralize it. And therefore these B cells, they divide, they become big, they secrete antibodies which are like magic bullets. They go attach to the pathogen and kill them. A story in little more in detail. So as you can see here, you can see the B cell. So these both of them are not being recognized. The particular B cell recognizes this bacteria. Once it recognizes, it gets the help from T cell. It becomes the plasma cell and the memory cell. Now I'll tell you what the memory cell does. Now the plasma cell starts making antibodies and releasing it like magic bullets. They bind bacteria. Now the macrophage is just waiting. It is a scavenger cell. And then once it sees the bacteria with the antibody bound on it, it just engulfs it. And the memory cells, so the infection is subsided, these memory cells keep moving in the body. So next time I see you, I know who you are. and therefore I'm going to kill you faster, much more stronger, and much more effectively. So this is the story about the B cell. Now, slowly we'll move on to the T cell. So the T cell, say the B cell can recognize the pathogen as it is and certain points, and it binds it. But the T cell cannot recognize the antigen by itself. As I told you, what happens in innate immunity? The virus enters, say the macrophage engulfs it, and then it comes to the adaptive immunity and tells the T cell there is a viral attack. I have already engulfed it. I'm showing you a part of it. So now you know that there is a viral attack. You better get activated. So the T cell looks at the portion of the virus that the macrophage shows, it gets activated. Once it gets activated, it then starts helping the B cell to become the plasma cell and produce antibodies, and it also helps the killer T cell. So, as I told you, the virus is taken up by the macrophage, and then the green portion of the virus is shown to the helper T cell. The helper T cell now produces certain signals, and then it activates the killer cell. Now, what does the killer cell do? The killer cell moves towards the viral infected cell, and then it attaches itself and tells you have to commit suicide. It does not do anything else. It tells the cell you have to die. And then it detaches, goes and attaches to another viral infected cell. And this particular cell slowly dies. Okay, so the killer cells take help from the helper T cells, recognize the viral infected cell, and then asks it to die. Similarly, it recognizes special components on the cancer cell and tells the cancer cell to die. In case it is an organ transplanted cell, it also tells it that you are non-self, you are not part of the body, and you have to die. So B cells will produce antibodies which bind the antigen, and this antigen antibody complex is engulfed by macrophages. T cells will look at viral infected cells, cancer cells, and then kill them. So their mechanisms are totally different. Now you have another type of cell called the natural killer cell. Now this cell is like the killer T cell, which recognizes a particular infected cell and then kills it. But the virus is very, very smart. If the immune system is smart, the virus is even more smarter. So what it does is, it does not allow the cell to tell the immune system that it is infected by a virus. So that the T cell cannot recognize it and kill it. So natural killer cells have a way of identifying such cells. So say for example, in the top panel, it is a healthy cell. The NK cell sees it, oh, you are a healthy cell, I need not kill you. But the targets or the viral infected cell, which tries to fool the immune system by not telling it, the natural killer cell senses it and it just attaches itself and it sends certain enzymes and the cell just dies. The same way as the killer T cell kills the viral infected cell. So this is another mechanism of killing viral infected cells. The last is a complement. A complement are small proteins which bind together only when they see a pathogen, and it forms a membrane attack complex. What does this complex do? It drills a hole into the bacteria. Once it drills a big hole there, all the contents come out. Water enters inside, there is osmotic pressure, the cell swells up and it bursts and the bacteria dies. So innate immunity, you have prevention of entry, you have physical barriers, you have chemical barriers, you have enzymes, then you have the process of phagocytosis and inflammation. And it also sends a message to the adaptive immunity. Adaptive immunity has B cells, which kill all pathogens which are in the bloodstream. You have T cells, the T helper cell, which helps the B cell, and it helps the killer cells. You have the killer T cells, which kill viral infected cells, cancer cells. You have natural killer cells, which kill the viral infected cells which fool the immune system, and you have complement which kills the bacteria. Imagine innate immunity and adaptive immunity have their own mechanisms. They work on their own, but again, they work in coordination. They talk to each other, they devise strategies and kill pathogens. Now, this is a very, very important slide. This will tell you what type of immunity have you developed. Now, it is of two types, active immunity and passive immunity. What do I mean by active immunity? Again, that is the immunity that you develop after you are exposed to a pathogen, whether it is live or dead. Depending on whether it is live or dead, it is further divided into natural and artificial. What do I mean by natural? You are exposed to a pathogen, you get an infection, your immune system responds by killing the pathogen, and memory cells are formed, and you are geared up for the subsequent exposure to the same pathogen. That means you are infected and then you get immunity. On the other hand, artificial active immunity is vaccination. What is vaccination? Vaccination is basically you're simulating a condition where the body is exposed to a dead pathogen. You are fooling the immune system by sending a dead pathogen. You do not get an infection, but the immune system sees the pathogen. It is non-self, it responds, and then you have memory cells. You are geared up because the immune system has seen the pathogen. It does not differentiate whether the pathogen is alive or dead. It does not matter. But in vaccination, you are exposed to a dead organism. You are not getting infected, but you are gearing up if it comes for the second time, because your memory cells are there. Your immune response is going to be quicker. It is going to be more stronger and more effective. So basically, vaccination is simulation of an exposure to a dead organism, but you are gearing up your immune system. Now, what is passive immunity? Passive immunity is you are borrowing immunity from somebody else. So you are passively transferring immunity from one individual to the other individual. Here again, you have natural and artificial. Natural is, say, a pregnant mother transports her antibodies through the placenta to the growing fetus. Once a child is born, these antibodies protect the newborn child from infections. The immune system of the newborn is weak, and therefore it uses the mother's antibodies to fight infection. Again, the mother starts feeding her baby. Through the milk also, some antibodies can enter the baby and they can protect the baby from infections. This is natural passive immunity. That means the antibodies, ready-made antibodies from the mother, protect the child. Now, on the other hand, we have artificial passive immunity, where we raise antibodies in animals, take their antibodies passively and use it to fight pathogens. But remember, in passive immunity, you are not activating your immune system. Temporarily, you're borrowing antibodies to fight the pathogen. Now, a typical example of artificial passive immunity is, say you have a snake bite. Now, you cannot wait for the body to produce antibodies because it is going to take 3 days or 4 days for the adaptive immunity to make antibodies. By then, you can die of a snake bite. So what they do is they take the venom proteins from the snake poison, they immunize animals, raise antibodies, and this is given as an anti-venom injection. So when there is a snake bite, immediately when you give antibodies, they recognize the venom proteins and neutralize it and save the individual from dying. So this is you're borrowing antibodies from another individual and you're utilizing it to neutralize it. Now, for diphtheria, for tetanus, snake bites, insect bites, you use ready-made antibodies that are available. So this tells you the type of immunity that we develop. A live pathogen, you have active immunity. Vaccination is a simulation process. Passive natural is from your mother. and artificial is from ready-made antibodies from animals. So this shows that natural is when you encounter a living pathogen. Vaccination when you encounter a dead pathogen. Passive immunity, transfer of antibodies from mother to child, and artificial is when you borrow antibodies made from some other animal to use. Now, what is herd immunity? This played a major role during COVID time. Now, see the first panel. You can see all the blue individuals are healthy and non-vaccinated. In the center, you can see a culprit. He's not vaccinated, but he's contagious. And there is only one vaccinated person, but it cannot help because this person spreads the infection to all the healthy people around, and finally all of them are infected. But if you are vaccinated, see the second panel. Almost all of them are vaccinated. In the center, you have a contagious person, but and one or two healthy individuals, but they are not able to contact the disease because finally this contagious person is not able to spread the infection because all of them are vaccinated. They are geared up, they can fight the infection, and the virus infection slowly starts coming down. So it was herd immunity, and that was developed due to vaccination, and that's how we could tide over COVID. Now, vaccination is nothing but immunotherapy, and all of us are mothers who would have given vaccinations to our newborn kids. You have hepatitis B, you have diphtheria, tetanus pertussis, that is DPT, then you have influenza virus, polio virus, MMR is measles, mumps, and rubella. You have chickenpox, then you have influenza, hepatitis A. Some are given as one shots, some are given as boosters. So the more the number of boosters, the more stronger your immune response will be. Then, as I told you, vaccines can be weakened viruses or bacteria. It can be killed bacteria. It can be the inactivated protein. Then you can have part of the protein. Then you have conjugate vaccines, DNA vaccines, and so on. So this is an example of COVID-19 vaccines because you are familiar with this. I made this slide. So you have mRNA vaccines like Moderna and the Pfizer. Then you have Covaxin, which is a dead or inactivated virus. Covishield is you take the DNA and then you put it in a viral vector, and the viral vector will transport the DNA into the body, and the DNA, makes proteins. So as I told you, the B cells only recognize certain projections on the virus. So if you give these spike proteins, which are projections, it is enough to protect the body from subsequent infections. So you can see the virus with these spike proteins. So you have live attenuated viruses, means attenuated is weakened virus. Then you have inactivated, either you heat kill it or treat it with chemicals. Then you have the outer projections or the spike proteins. These are enough. Once they are exposed to this, your B cells will recognize it and you will elicit an immune response. Then you have the genes which are transferred. So you have mRNA and DNA, and then they are transferred, and the body makes antibodies against these spike proteins. Now, we looked at vaccinations. Now we'll move on to cancer immunotherapy. Now, what happens during a normal response? So your T cells will kill the tumor cells once they identified. Once an infection is done, you know that during infection, the B cells are very active. They are actually hyperactive. T cells are hyperactive. So you have to control them. So normally we have a regulatory mechanism to slowly suppress the T cells and the B cells. So the cancer cells are very, very smart. They have taken advantage of this, and what they do is they try to suppress the immune system so that they can survive and divide. Okay, so there is a new therapy called the blockade therapy, where you block these molecules, the one you see in brown and yellow. They block it, and they and this will reactivate the T cells, which can now kill the tumor cells. So this is called the blockade therapy. Now we know that cancer therapy is either chemotherapy or radiation therapy. And we know chemotherapy not only kills healthy cells, it also kills cancer cells. And there are a lot of side effects. Radiation therapy, though it is focused on a particular organ, all the healthy tissue around it is also damaged. So the best way would be to activate your own immune system to kill the cancer cells. So this is one method. The other method is the CAR T cell therapy. You are developing super soldiers. So you take your own T cells, so they are self, so the body will not mind accepting it. You remove the T cells from the body, you engineer them so they develop new antenna which can recognize the cancer cells. The ones in green are already present there, but the ones in red are the new ones that you have engineered. They are all equipped. It is called the chimeric antigen receptor. You introduce these engineered CAR T cells into your body again. They are geared up, they are super soldiers, and they go and recognize the cancer cell and kill it. So the blockade therapy and the CAR T cell therapy are activating your own immune system to fight cancer. Then you have monoclonal antibody therapy. So as I told you, you can make antibodies in the lab. And once you make antibodies, you attach an anti-cancer drug to it. And I told you, these antibodies recognize certain components on the surface of the cancer cell. And once it is tagged with the drug is in the close vicinity of the cancer cell, and the cancer cell can be killed. So these are some of the latest immunotherapies for treating cancer. Now I have spoken about the immune system, I've spoken about innate immunity, I've spoken about B cells, T cells, natural killer cells to fight and elicit an immune response. Then we spoke about immunotherapy, vaccinations, cancer therapy, where we looked at blockade therapy, we looked at CAR T cell therapy and monoclonal antibodies. Now, immune system can also help in diagnosis. Now, say you have typhoid. How does a doctor know that you have typhoid? Okay, clinical symptoms are in one, but they have to identify the organism. So what do? They either recognize the pathogen by, or they recognize the antibodies that are formed to that particular pathogen. Either way, antigen-antibody interaction is required for diagnosis. You have a wide variety. So your rapid antigen test, RT-PCR, your Vidal test, your pregnancy test, all these tests are for diagnosis. So you can detect the antigen or you can detect the antibody. So we have looked at immune responses, we have looked at immunotherapy, we have looked at immunodiagnosis. So the takeaway message from my talk today is healthy self means heal thyself with the power of your own immune system. You split healthy into heal thyself. You are healing yourself with the power of your own immune system. Please give your immune system a fighting chance. It is capable of protecting you from infections. The problem is we do not have the time. You feel you should get rid of your infections within minutes, within hours, but it is not possible. Innate immunity can attack immediately, but adaptive immunity requires some time. Please give your immune system some time so that it can fight against pathogens. And to have a strong immune system, you should have a healthy lifestyle. A healthy lifestyle will give you a strong immune system, and a strong immune system will protect you against all pathogens and infections. And this is how you should understand your immune system to fight your pathogens. Thank you for a patient listening. And I think I should thank the Alumni Association for giving me an opportunity to address all of you. My specific thanks to Vasavi and Tinku for arranging this lecture. And I think I should thank Anuradha of my batch for putting all these things together. And I thank all my teachers who have attended this lecture, Sister Preside, Mrs. Lakshmi, and all my classmates and anites for attending the lecture, and hope you enjoyed it. Thank you.